Assalamu alaikum. Welcome again to online lecture class. Today I discuss about the last part of cell biology, and this cell biology contain lecture contain the different genetical term and mitosis, meiosis, cell cycle. I know that you those things are well known for you all, but I am taking revision and. At first you have to know the chromosome, and you already know inside the nucleus different rod-like structure present in network pattern, that is termed as chromosome and made up of DNA and protein. Total 46 number of chromosome in the body, 44 are autosome and 2 are sex chromosome. In case of male it is termed as spermatozoa, and in case of female it is known as ova. And for when it remains as 46 chromosome or total chromosome number, it is termed as diploid, and when it remains as 23, that is termed as haploid. And two arm of chromosome, that is short arm chromosome, here showing actually diploid number chromosome, because here pairing of chromosome present and attached with a central centromere, and this position of centromere may be different. It may be center or subcenter or near the end. If it is near the end, it is termed as telocentric. When it is remain in the center, it is metacentric. When nearer the end, it is submetacentric, and close to the end, that is acrocentric. And you see that diploid number of chromosome, when a single chromosome, that means one single short arm, one single long arm present attached with centromere, that is termed as haploid. And when two pair, mainly here showing the two pair chromosome, that is diploid. What is gene? Genes are actually a unit of heredity. That means it controls the characteristics of an offspring. Will have be transmitted information sequence, and it transmitted to uh, from father to daughter cells, or parent, from parents to daughter cell. And what is DNA? DNA, you know, a double helical structure, and united by hydrogen bond, and few nitrogen base also present, that is adenine, thymine, or cytosine, guanine. Those are well known for you all. And in case of RNA, it is a single strand, and it has ribose unit and phosphate unit, and nitrogenous base are contained here, that is adenine, guanine, cytosine, or uracil. What do you mean by homologous chromosome? Homologous chromosomes means paired identical chromosome. That means having a paternal and maternal member. Locus. Locus means position. The position of gene is known as locus. Allele. Allele means the genes occupying the identical loci in a pair of homologous chromosome. That means genes when occupying in a identical loci and remains as a pair of homologous chromosome, termed as allele. Homologous. Homologous means two parental genes are identical for a particular character are known as homologous gene. Like father and mother hair color is black, it's a identical character, and it is also homologous. But if it remain as a brown hair color and golden hair color in case of foreign parents, that means it is heterozygous. Heterozygous when two parental pair gene are contain different character, that is heterozygous gene. But if it is identical character for a particular character, that is homozygous gene. Karyotyping. Karyotyping is the test to identify or and evaluate the size, shape, and number of chromosome. Genome. Complete set of gene DNA is known as genome. It is contain all of the information needed to build and maintain that organism. It is haploid in gamete and diploid in the somatic cell. That means the body cell, all cells remain as diploid, and only germ cells remain as haploid. Genotype. Genotypes means genetic constitutions of the individuals, which is fixed at the time of fertilization. In case of tall, it is represent as the large letter TT or large letter T, a small letter t. Phenotype means the biochemical expression of outside of the body physical character is known as phenotype. That means inner genetic character is known as genotype, and outer biochemical expression or physiological character are known as phenotype. Genetic engineering, that is manipulation of DNA to produce new type of organism, usually by inserting or deleting the genes. Cloning, the use of DNA multiplication procedure to produce multiple copies of a single gene or segment of DNA. Now cell division, I describe it within a short time. That is two type of cell division present in our body, that is mitosis, meiosis, you already know. And all somatic cell of the body and immature germ cells can produce mitosis cell division, and only germ mature germ cell uh, perform the meiosis cell division, and the process of mitosis is only 1 to 2 hours. In case of mitosis process, it has a different phase. That means at first prophase, and in case of prophase, thickening of the chromosome material. Thereafter, in case of metaphase, uh, here showing the thickening of the chromosome. In case of metaphase, mitotic spindle form, and centrosome are divided into two centriole, and centriole attached with the mitotic spindle. Thereafter, in case of anaphase, the centriole and chromosomes divide into two form, and moving towards the opposite pole. And in case of anaphase, two cells are divide into the daughter cells. So here separate in separate form, that is in case of prophase, it's thickening of the chromosomes, disappearance of the nuclear membrane and nucleolus. In case of metaphase, mitotic spindles appear, and centromere attached with the mitotic spindle, and here showing centrioles are remain in the two opposite pole. Here two pair of chromosomes that are moving to the opposite side. In case of telophase, you see the reappearance of the nucleolus and nuclear membrane, and daughter cells of the chromosomes are collected in each end of the spindle. Now hazard, what are the hazard happen in the mitosis? That means non-disjunction, anaphase leg, and isochromosomes. And during telophase, polyploidy can happen. And I will discuss about those things later. At first, I want to discuss the meiosis process. Meiosis occurs in two stages, that is meiosis 1 and meiosis 2. All are happen as like mitosis, prophase 1, metaphase 1, thereafter telophase 1, anaphase 1, telophase 1. In case of meiosis 2 stage, prophase 2, metaphase 2, thereafter anaphase 2, telophase 2. Those are sequentially event happen. In case of mitosis, uh, in case of mitosis process, one parent cell form two daughter cells, but and chromosome number remains equal. That means 46, 46. But in case of meiosis process, one cell divided into four daughter cells, and chromosomes remains half of the parent cell. That means 46 convert into 23, 23, and The in case of mitosis occur in the all somatic cell of the body, and immature germ cell, that means initial phase of germ cells, and in case of meiosis process happen in the mature germ cells. Now anaphase lag. What do you mean by anaphase lag? That means I already told the different hazard present in the mitosis: non-disjunction, anaphase lag, and isochromosome. So anaphase lag actually a consequence of event during cell division where sister chromatid not properly separate from each other, and improper spindle formation occur there. That means when parent cell divide in the mitosis process, that means two chromosome paired two chromosomes are separated and goes towards the opposite pole. In that time, separation do not happen completely or properly, and the spindle formation also is defective. So there is a complication occur, that is anaphase lag. It is the one of the many causes of aneuploidy, and this event can occur during mitosis, meiosis, both process. So what do you mean by aneuploidy? Aneuploidy means extra or missing chromosomes. That means when divide in a non-disjunction process or division process, the cells, parent cells divide into two daughter cells, and two daughter cells actually contain equal number of chromosomes, but during division few daughter cells contain extra chromosome and few daughter cell miss that chromosome. So there may be dissimilarity, extra chromosome or missing chromosome. If it is extra chromosome, that is term 47, and missing chromosome 45 number of chromosome. Aneuploidy occur two cases: non-disjunction, anaphase lag. I already tell that anaphase lag shows the improper mitotic spindle formation or improper division of chromosomes, and it actually process of the chromosome moving is too slowly occurred in anaphase lag. That means during anaphase, two chromosomes when separated to opposite pole, the process of separation is too slow occur, and that wise the chromosomes cannot separate properly. And in case of non-disjunction, paired chromosome both go to the same pole in meiosis instead of opposite pole. I'll discuss later. Here showing non-disjunction 46 chromosome 23 23. That means here showing normal in haploid division, but in case of here meiosis 2, here 46 to 23 it is meiosis 1, and 23 when divide again in meiosis 2 process for 24 in one chromosome, that means extra copy of chromosome, and here contain 22. So here non-disjunction happen in meiosis 2 stage, but here 46 divided into 22 and 23, that means here showing meiosis 1 stage, non-disjunction, one chromosome missing here, one chromosome, that means totally miss one chromosome here, but it is normal 23. Again when division in second meiotic division 22 22 22 and 23 remains 23 one daughter cells and another daughter cell contain 22. That means here also one chromosome missing, and here actually anaphase lag in meiosis 1 and meiosis 2. That means delay of the or slowing of the chromosome separation occur, that was uh, anaphase lag occur. In case of monosomy after fertilization, it happened. Monosomy means missing the chromosome number. I already described those things: non-disjunction, anaphase lag. Now isochromosome. What do you mean by isochromosome? We know that the in case of chromosome, two short chromosome, two long arm chromosome present and attached by centromere, but and equal distribution of short arm long arm present in a chromosome. That means in here showing haploid number of chromosome short arm long arm, and here showing diploid number of chromosome short arm long arm, but in case of isochromosome, during division of cell, the short arm remains in same side, long arm remains in same side. That means two green color showing short arm and two violet color or purple color showing the long arm. The long arm remains in the same chromosome and short arm remains in same chromosome. It is isochromosome. It is cytogenic variant of Turner syndrome. We'll know Turner syndrome later. So unbalanced structural abnormality in which the arm of the chromosomes are mirror image of each other, like short arm and long arm chromosomes remains in same in the same chromosome. That means in normal case one short arm, one long arm, but in isochromosome there is two short arm or two long arm. That is present in case of partial monosomy, or maybe a few genes are lost. Now, prophase 1 in case of meiosis. Meiosis prophase 1 is important for exam. Otherwise, I already discuss the meiosis 1 prophase. Prophase 1, few stages present, that is leptotin, zygotin, pachytin, diplotin, diakinesis. In case of leptotin, you remember one sentence, that is long, thin, thread-like chromosome and beaded centromere appear along their length. And in case of zygotin, two chromosomes, similar characteristics, that is termed as homologous, are remains in a same site or same area, that is termed as synapsis. Synapsis formation occur in zygotin, and long threaded chromosome occur in leptotin. Thereafter, pachytin. Pachytin, homologous chromosomes actually paired form are tetrad formation occur. That means total 4 arm of chromosome, homologous chromosome, are remain in same area and form tetrad. And in case of diplotin, in two homologous chromosome crossing over occur of non-sister chromatid. That means crossing over means crisscross manner, two chromosomes break their arm and attached with the non-sister chromatid. And this process is known as chiasmata formation, that is occurring diplotin. It is important MCQ question, that is chiasmata formation and crossing over where happened, that is diplotin stage. And few enzyme cells to cut the chromosome and ligate in another non-sister chromatid, that is endonuclease and ligase. Endonuclease actually cut the arm of the chromosomes, and ligase helps to attach that cut portion of the chromosome to non-sister chromatid. Those two are enzymes. And diakinesis process, separation complete of the chromosome and disappearance of the nuclear membrane, nucleolus occur. That all process happen in the prophase 1. Here picture actually fade, but try to show you that the long thread-like chromosomes in present the leptotin stage, zygotin stage, the two chromosomes are remains as paired homologous chromosome, that is synapsis. Tetrad formation occur in the pachytin stage, that means 4 arm of the chromosome in attached here. It is not attached, it is remain same area actually. And thereafter non-sister chromatid are crossing over occur, and crossing over and chiasmata formation occur in diplotin stage. Thereafter completion and genetic information transmission complete in diakinesis process. Nuclear membrane are disappearance, and nucleolus also disappearance in prophase 1 stage. Difference between mitosis meiosis important, and you already know. Now cell cycle. Cell cycle are the actually moving, or it is actually process that happened mitosis and interface in same time. That is at first It is actually event, event that two process happened continuously, that is mitosis and interphase. At first, the mitosis phase, one cell divided into two daughter cells. Thereafter, in case of interphase, there are few stages present, that is G0, Z1 S phase and G2 phase. Z0 actually remains dormant in our body. Three type of cell present: permanent cell, labile cell, and stable cells. Permanent cells are neuron, cardiac muscles. That permanent cells means when it damage occur, repair does not possible. That means totally permanently present in our body. And in case of labile and stable cells, of regeneration process can be happen if it is damaged. Now, in case of Z0 phase, only permanent cells remain as dormant. They do not undergo further division or interphase. But in case of stable labile cells, they undergo the further stage. That means in case of G1 phase, gap between mitosis and DNA replication phase, that means pre-synthesis phase of DNA, and the DNA synthesis occur in S phase, and post-synthetic DNA phase, that means between DNA replication and next mitosis stage, that is G2 phase. G1 phase is decision making phase. This period active RNA and protein synthesis occur. Total duration is 25 hour. In case of S phase, DNA replication occur, that means synthetic phase. Here histosynthesis occur, and duration is 8 hour. G2 phase 2.5 to 3 hour. Here in this phase, protein required for mitosis are accumulated, and after the G2 phase, the mitosis process again start. I already told that G0 phase, here permanent cell remain dormant, and only stable cell labile cells undergo further G1S G2 phase. So thank you all.